is on aquatic microbiology, specifically on the importance and regulation of composition and community dynamics in marine bacterioplankton. Lasse has lately focused on nitrogen fixation with extra emphasis on heterotrophic nitrogen fixation. Previous work of his has been on viruses and larger organisms like teniforms, comb jellies, and fish larvae. Lasse, I thank you for joining us today and welcome you to our session this afternoon.
Okay, great, excellent. Thank you very much. And good afternoon, everyone. So I'm just gonna share my screen in a minute. But before doing that, please feel free to interrupt me during the talk. I'm not gonna see if you have any written questions. We can take those afterwards, but just interrupt me if you wanna shoot with a question. So let's see here. Rachel, I hope you all see my screen now. Rachel told me, just minimize this. Rachel told me that you just heard the talks by Jonathan Sayre and Bess Ward, and that's great because then you know a bit about this topic. So I will be talking about the nitrogen fixation by putative heterotrophic bacteria, and the focus will be on coastal and estuarine waters. You basically also then know what is shown here. Nitrogen gas is the most common element in the atmosphere, and it readily dissolves in seawater. So there's always plenty of N2 in seawater. The nitrogen fixers, they are able to split the triple bond in the nitrogen and convert it to a usable form of nitrogen in the cells and for the food web. And remember that this process is exclusively carried out by prokaryotes and that it's an anaerobic process. It's also a very significant process in tropical and subtropical marine regions. It has many times been estimated that maybe up to about half of the new plankton production is due to this import of nitrogen through nitrogen fixation. In temperate waters, nitrogen fixation have been believed to be insignificant, and that also goes for estuaries in general. One major exception is the Baltic Sea, where it's been known for many years that there's extensive nitrogen fixation. Historically, when you talk about marine nitrogen fixation, everyone has thought about cyanobacteria and exclusively cyanobacteria. But there is accumulating evidence that there's also a role for non-cyanobacteria, which I will also call heterotrophic bacteria. And that's what I'll try to highlight to you during this talk. So it's been known for quite some years that the marine non-cyanobacteria, they are diverse and they're found pretty much everywhere. This is an old phylogenetic tree back from 2010. It is based on about 2,500 NIF-H, that's nitrogenase genes, amplified from marine and estuarine plankton. And the round balls you see here are where they are found in open marine waters also. If we did this tree today, there would be many, many, many more sequences, but the picture would more or less remain the same. And that is that the diazostrophs and the non-cyanobacterial diazostrophs are really diverse. And also they are found pretty much everywhere we look in marine waters. It's also well known that non-cyanobacteria, they often account for a main fraction of the nitrogenous gene pool in marine surface waters. And this, what you see here, is an example of a study. It was by my previous PhD student, Hannah Farnelid. And What she did was to amplify nitrogenase genes from surface waters from around the globe and sequence them. And what you should look at here on these pies are just the colors. In green, you have cyanobacteria, and all the other colors are heterotrophic, putative heterotrophic groups. So this tells us that actually, even in surface waters, in many places, most of the nitrogenous genes come from heterotrophic bacteria, not from cyanobacteria. Now, that does not mean that the heterotrophic bacteria are responsible for most of the nitrogen fixation, because these are only genes in the DNA, and we don't know whether they are expressed. But still, when we look through the literature, there is now convincing evidence for non-cyanobacterial nitrogen fixation, both in photic waters and in aphotic marine waters. But still, their quantitative importance remain unknown. And related to that are also that since rates are really low often, we still have a number of issues related to methodology. It's really difficult to measure nitrogen fixation when rates are really low. Still, if we, for instance, do a back of the envelope calculations on the non-cyanobacterial nitrogen fixation rates down in the deep dark sea, as my previous postdoc Ma Benavides did, if you do this back of the envelope calculation on that, you will see that this fixation could potentially be of global budgetary significance. And that's because even though rates are really low, the volume of the dark ocean is so huge. So if you multiply those two numbers, it will be a very significant number. So currently, with respect to the non-cyanobacterial diazostrophs, we are at the state where we know that they are widespread. We know that they are occasionally active, but still we lack a mechanistic understanding of their ecology, and we don't really know about their quantitative importance. And that goes actually for surface waters as well as for the deep sea. Now, if we look at coastal waters compared to the open ocean, there are some marked differences. In the coastal ocean, we will often have higher levels of dissolved and particulate organic matter. We will have, in general, higher nutrient levels, often occasional nitrogen limitation. Sometimes we will have low or at least reduced oxygen conditions. And the question is whether such coastal waters then, wouldn't they be more suitable for nitrogenation by non-cyanobacteria than, for instance, the open ocean waters? Well, that's at least what we have speculated. And I'm going to show you an example of a study from some of our coastal waters. It was my previous PhD student, Mikkel Bensantilia, that did this. He, over a year, he sampled surface waters from this Roskilde Fjord on Zealand. This is quite a eutrophic system, very shallow, less than 5 meters deep. It's a very stable system and very nutrient rich. Then he also sampled this station in the Great Belt. It's much deeper, 35 meters, and it has strong changing currents. As you can see, all the water from the Baltic Sea comes out through the Danish Straits, and this is one of the straits where the water comes out. So it's very influenced by an outflow from the Baltic Sea. So he looked at these two contrasting environments over a full year. And what you see here is nitrogen fixation measured by 15N incorporation, and you have the full year. And you have Roskilde Fjord on top and Great Belt down below. And what's striking here is that you can see intense and high rates of nitrogen fixation. These are very high rates. It's comparable to in the Baltic Sea. What we can also note is that we have a spring peak in both places, and then we have a summer peak in the Great Belt. We can also note that fixation measured in the dark, it's the dark spots here, is actually a significant proportion of the total nitrogen fixation in the two spring peaks, but not in the summer peak. There's basically no dark fixation here. Mikkel then tried to find statistical correlations of these nitrogen fixation rates to all kinds of parameters that he measured, but he could only find one, and that was that nitrogen fixation in the Roskilde Fjord. It was correlated with dissolved organic carbon, as well as with bacterial production and abundance. And I can get back to that. Then he used molecular methods to analyze the expressed nitrogenase genes. So he extracted RNA and then did sequencing of the expressed NiFH genes. And that's what you see here in these heat maps. On top, you see the measured nitrogen fixation rates, and then you see the heat maps below. So these are dominant types of nitrogenase genes, the period. And when looking at that, you can see differences between the two environments. You can see that in Roskilde Fjord, it was mainly heterotrophic bacteria expressing these genes, but also Eucin A. Eucin A was this single-celled cyanobacterium that lives in a symbiosis with a small phytoplankton that Jonathan Sayre has told you a lot about. So this one was also found in Roskilde Fjord. Then in the Great Belt, the composition of the active nitrogen fixers were much more diverse, consisting of heterotrophic bacteria, Eucin A, and then also this cyanobacterium called Anabaena. And Anabaena is one of the typical nitrogen fixers from the Baltic Sea. It contains heterocysts, it's a filamentous form, and so it was also very prevalent. Then what can be done from these kinds of data is that I don't know if you've heard about quantitative PCR, but it's a way you can design primers and probes so you can actually quantify the abundance of specific types of nitrogenase genes in the samples. And Mikkel did that. And what that told us is that the nitrogen fixation peak in Roskilde Fjord in spring and in the Great Belt also in spring was dominated by heterotrophic bacteria and Eucin A. And that's also consistent with that the nitrogen fixation took place both in the light and in the dark, and it was in the less than 10 micrometer size fraction. And that makes sense because these cells are all small. Now the summer peak here, that was carried out by heterocystic cyanobacteria and Eucin A, and that was consistent with most of the fixation actually was in the dark, in the light, and also that most of it took place in the larger than 10 micrometer fraction. So by combining these measurements as well as the molecular data, he could sort of put names on who were the active ones in this environment. So the main conclusions from this study was that the nitrogen fixation in Danish coastal surface waters is significant but highly variable. This was striking at the time because it had been always assumed that nitrogen fixation in Danish waters was zero, which is not the case. Another important insight was that the relative importance of heterotrophic, photoheterotrophic, that refers to Eucin A, and heterocystic cyanobacteria differed a lot in time and space. So this means that this study and also several other recent studies have shown that non-cyanobacteria, they can be important nitrogen fixing organisms in coastal and estuarine waters. Still, we have a relatively poor understanding of their ecology and also of the environmental factors that govern their nitrogen fixation. Although Mikkel, for instance, found this coupling between nitrogen fixation in Roskilde Fjord and a coupling to the amount of dissolved organic carbon being present. So apparently that was a factor that looked important in this study. But in general, if we if we look through the literature and think of the main factors that control nitrogen fixation by marine non-cyanobacteria, that would be the presence of oxygen, because nitrogen fixation is an anaerobic process. The presence of readily utilizable fixed N, that refers, for instance, to ammonium and nitrate, and then energy availability. It's expensive to carry out nitrogen fixation, and it's expensive to protect yourself against oxygen. So these looks like they are the main factors. We've then looked further into these factors, and we have done that through our work with isolates. We have used dilution to extinction techniques, as shown here, to obtain isolates of heterotrophic Diazostrophs from the Baltic Sea. We've done this several times, and every time we have, when we have acquired the isolates, we have used this quantitative PCR to go back into the environment and to demonstrate that they are really found out in the environment at significant concentrations, or that they are important in when we do nitrogenous gene sequencing, we find them out there. So that means that we have an argument for that. We think that the isolates that we work with, that they are environmentally relevant. And that's always important when you work with isolates, because you risk always to get the criticism that they are not relevant out there, that they're just bugs that come up in culture. But ours are environmentally relevant. We've done some work on these, and one of the first things we did to genome sequencing of our, and he found that they were surprisingly metabolically versatile. For instance, they had a lot of genes involved in degradation of aromatic compounds. That's typically something from oil. He found that there were some examples where they were actually able to do nitrogen fixation at quite high oxygen and at quite high ammonium levels. That's very, what should I say, counterintuitive to find that. We did not expect that. But in general, we could see that nitrogen fixation was reduced at high levels of oxygen. That's also what we would expect. If we take this information and then we think about life in the marine water column for a small putative nitrogen fixed heterotrophic bacterium, the water column is generally nutrient poor and is generally oxic. So we would assume that these bacteria, in order to carry out nitrogen fixation, they would be looking for some kind of loci with low oxygen concentrations that would also be rich in energy. So where do we have that in the marine water column? Well, one suggestion would be particles. There are all kinds of particles in a marine water column. Just some examples are shown here. So such particles, they would be loci where lots of energy would be available for microbial respiration and then possibly for nitrogen fixation. And if these particles are colonized by bacteria, well, then microbial respiration could generate local microoxic conditions. So we posed the hypothesis and have earlier highlighted that particles could be a site for heterotrophic nitrogen fixation. This was not our idea. It was highlighted back in the 80s by Hans Perl and collaborators. And on marine snow particles, we often see that they can have low oxygen conditions within them or even anaerobic conditions. This is an example from a study from 2015 from Helle Ploug's group. Here they looked at nodularia aggregates as shown here. They are actually quite compact aggregates, but nevertheless, when they measured oxygen concentration throughout the aggregates, as you can see here, they contained anaerobic zones within them. And those anaerobic zones would be dependent on how much oxygen was available outside in the surrounding water. So you can see if there's a full saturation of oxygen in the water, the zone will be smaller, and if oxygen concentrations are reduced in the surrounding waters, the anaerobic zone will be larger. Now, this has also been shown for a number of other types of marine particles, where it's been shown that dependent on their size and therefore the associated respiration on them, that will sort of regulate the extent of low oxygen or anaerobic zones within them. But it's been found then in many types of particles. We've then looked closer into this question with particles, and my previous master students Jeppe Pedersen and my postdoc Dennis Bumbar, they did some experiments related to that. They looked at particle-associated nitrogen fixation and diazotrophic succession on surfaces. And first, they did a very simple experiment, which is shown here. They took water from Roskilde Fjord, so the fjord that I started out with showing you, and then they enriched the water with particles from the water itself. So they did a 10-fold concentration of the particles. And they could see, yes, the presence of particles that stimulates nitrogen fixation. They also did another interesting thing, and that was that they took a little bit of surface sediment and added it to the incubation, and that led to an almost 100-fold increase in nitrogen fixation. This is interesting because it indicates that resuspension of surface sediment would occur in a lot of shallow areas, that potential to lead to a manifold increase in pelagic nitrogen fixation. That's very interesting. It's a slightly different story, so we'll leave it there. But anyway, it's interesting. But the take home from this is that, yes, it seems that particles, they stimulate nitrogen fixation. This is just, if you're interested in this, I can recommend you this paper that's on the same topic, and it's an excellent paper. Now, we have also tried to approach this particle-associated issue with some of our isolates. So my previous postdoc, Ryan Pearl, he looked into our Pseudomonas isolate and looked at oxygen conditions and surface colonization with respect to that isolate. And he found that planktonic cells can fix of Pseudomonas in culture can fix nitrogen at up to 54 micromolars of oxygen. So here you have nitrogen fixation, here you have oxygen. So it means that they can actually fix nitrogen even though quite some oxygen is present. He also tried to add different types of surfaces or particles to the cultures, but he was not able to stimulate nitrogen fixation under aerobic conditions. Only if he took the isolate in culture and forced it onto filters, for instance, so did some kind of artificial aggregation down on a filter, then he could stimulate nitrogen fixation under aerobic conditions. We were thinking about how to interpret those results. But at least this whole scenario, it highlights the significant oxygen problem that heterotrophic diazostrophs, they will have in the oxic water column. And these results also pose the question that maybe an initial and extensive surface-associated growth of other microbes are needed prior to this fixation by diazostrophs. So maybe there needs to be an existing microbial consortium on the surface before the diazostrophs can come in and do their nitrogen fixation. Related to that question, we did some other things. Jeppe and Dennis, they looked at surface colonization and the proliferation of diazostrophs. And they did this by, here out in this picture, you can see we have some small filter holders with filters on them with a small bit of substrate on it. They put these filter holders in Roskilde Fjord water for 400 hours and then examined the colonization of these filters, but also did sequencing. So did sequencing of the 16S RNA genes to find out who are the bacteria on these filters, and also did nitrogenous gene amplicon sequencing to find out about the diazostrophs specifically. In this graph down here, you can see over time, 400 hours, and you can see how the number of cells on the filters increase. So a biofilm is forming on the filters, and that could also be directly observed in the microscope. So you can see microcolonies forming here. They did this 16S sequencing, and that's what is shown here. This is quite a busy slide, but I'll try to walk you through it. So when you do the 16S sequencing, you get a number of operational taxonomic units with a relative abundance. You can then convert that relative abundance to cell numbers by multiplying with the actual number of cells in the water or actual number on the filter. And you do that because you have to take into account that the cell abundance that change a lot over time. So here you have a number of cells per milliliter. In the middle here, you have the taxonomy of the predominant operational taxonomic units, and you have whether they were associated or related to known free-living bacteria in the database or known attached bacteria in the database. And if we look at the community in the water surrounding these filters, we can see that initially in the experiment, all of the bacteria were related to free-living bacteria, which are known as free-living. We can see later in the experiment down here, we can see that it becomes a mix, so that the bacterial community consists of bacteria related to free-living as well as some related to attached bacteria. On the filters, it was a bit of a different case. We saw a pronounced shift to very abundant OTUs related to taxa that are known to thrive on surfaces. So these are probably specialists that really like to live on this surface. And this succession that we observe, it likely reflects how the environmental conditions on these filters change over time. If we then look at the diazostrophs, they could not be amplified until late in the experiment. In a single sample at 80 hours, one sample could be amplified, but not then before 300 and 400 hours. We can also note that it has a very low diversity. It's single phylotypes here that dominate. And if we look, they are secondary or tertiary colonizers of particulate matter. So they come in late. It's the pioneering species. So the first bacteria that colonize a surface, they grow, they respire, and thereby they facilitate the later conditions that are suitable for nitrogen fixation. For instance, they produce the suboxic or anoxic conditions on a surface. So if we overall take a look at what I've told you until now, we know that the non-cyanobacterial diazostrophs, they are present, they are occasionally active in coastal temperate waters. We know that in some cases, researchers have been able to demonstrate that they carry out fixation, but it's at low rates. And there is always the inherent problem that their activity is really difficult to distinguish from cyanobacterial activity. Even in environments where there'll be very few cyanobacteria, if there's just one in the sample, it may confuse the result. I hope I've been able to show you that by combining fieldwork, isolate, and experimental work, we're slowly gaining insight into the ecology of these bacteria. In my opinion, particles in the marine water, it remains the most suitable loci for pelagic nitrogen fixation by these non-cyanobacteria. But actually, hardcore data are needed. And it's very difficult because these bacteria are found at so low concentrations and their activities are so low. Still, we're continuing on this work. And my current postdoc, Subhendu Chakraborty, he's a modeler. So he has made a mathematical model of these unicellular heterotrophic bacteria growing on a sinking marine particle. And his work documents that indeed these low oxygen conditions are generated over time, and that indeed this can provide a window of opportunity for nitrogen fixation by heterotrophic bacteria. And also in his model, he's been able to estimate rates, and the rates that he estimates, they are actually similar to the rates that people have measured in the dark ocean. And the rates in the dark ocean would putatively be originate from heterotrophic bacteria. So that's a very, very nice confirmation there. And we have recently submitted his work. We've also done something else, and that is we have done experiments with our isolates, and we have looked in the marine databases of whole genomes of heterotrophic bacteria. And by doing that, we have been able to demonstrate that these heterotrophic bacteria, many of them have motility and they can do chemotaxis. It's much more widespread among these diazotrophic species than it is among marine bacteria in general. And this is important because if we hypothesize that marine particles are the main sites for their nitrogen fixation, well, then they have to be able to find a particle. You can't just hang out in the water column and wait to be hit from above by a particle. You have to find it and swim down and colonize it. And this is what my student has identified. This is Søren Hellstrøm who did this work. Overall, back in 2007, we wrote that there's a need for combining rate measurements and molecular analysis of field samples with cultivation in order to clarify the ecology of non-cyanobacteria and their contribution to marine nitrogen fixation on local and global scales. Well, that's 4 or 5 years ago, and I would say still today, this is still valid. We have moved forward, but there's still a long way to go before we have a more comprehensive understanding of the importance of these bacteria. So all of this work was financed by the several projects from the Danish Council for Independent Research, and then an EU project named Blueprint. And these are all the people that did the work. And of course, a number of collaborators also were involved in this. Finally, I would like to advertise that I don't know exactly what this presentation will be used for, but anyway, I'm looking for a PhD student and a postdoc starting my group from spring. So if you hear about anyone, just let me know. And that was all, and I'm happy